I am going to give you a sheet here for you to check off what things you have done that fit into that standards category. So bring this back to the test on Thursday, and this will kind of give you a sense of which of those rating categories you're falling into. Are you going into the default category where all of your tests are going to count? Are you going to go into the category where your tests are not going to count as much? So you should pretty much be done with chapter 1. I've put later due dates on the chapter 2 stuff. So at the bottom it says looking ahead to chapter 2. Those aren't things that you have to have done right now, but they are things that you're going to need to get done eventually. So you got to make sure you are keeping that on your radar. On Canvas, I have started to put complete and non-complete things. And the idea behind that is just to let you know when I've noticed that you have completed a category. Because remember, for the standards, I'm not so much concerned with exactly what your percentage is. You have to meet a minimum of 80% on the video quizzes, but just that you've done it. So if you've done 2 of the video quizzes and if you've gotten at least 80%, then I've got that checked off. Now that's one of those standards categories. Maybe you don't want to have an A, you're okay with a B, you don't want to work that card. So if you've done 2, I've got that checked. But if you've done 3, I've checked both the 2 and the 3. Okay, so in order to fit into the highest category, if you're aiming for an A, you want to have 3 videos done for each chapter. So steroids make hormones, not all of them, but some of the most important ones. So testosterone, estrogen, progesterone, those all come from this basic molecule, the ones that as sex hormones. And then there are other hormones too. And we will talk about cortisol later in the semester because that one's really important with digestion. And then it also can transport food. Okay, so going back to these things here, these are long chains, and we said these zigzags here just have a bunch of carbons on them. And the word here tells us it's some kind of an acid that comes from this functional group here. So this functional group has 2 oxygens instead of just 1. So what do we call in general these things that have a long chain of carbon and then an acid? Say again? Put them together, it would be a triglyceride because there's 3 of them, but one individual is a fatty acid. So by themselves, they're fatty acids. Okay, and then we talked about how if they have no double bonds, they are saturated. And if they do have double bonds, we call them unsaturated. And then there are some different kinds. You've probably heard of trans fats. We don't usually say cis fats, but that's the alternative to trans fats. So this one is cis because these lines are both coming down. And so we've got this little bend in our chain, a kink in our chain. So there are a couple of videos that I have posted the quizzes for. The videos were already posted that talk a little bit about trans fats. There's one that talks about healthy fats and one that talks about biomolecules. I think the one on biomolecules would be a really good review for your test on Thursday. So you might look at that, but they describe the difference between these 2 kinds of fats. The main thing we want to remember is what a fatty acid looks like. So if you look at old exams, you'll be able to see, sometimes I'll just pick out one of each of these pictures and say, what kind of molecule is this? Be able to tell the difference between saturated and unsaturated. So saturated means there's no double bonds, except for the ones to the oxygen. There's no carbon-carbon double bonds. And unsaturated means there is a double bond. And then the saturated are not so good for you. Trans are not so good for you. The cis are better for you. All right, and then the last one we didn't really talk a whole lot about, that's up in this upper right-hand corner here. And so it kind of looks like a triglyceride because it's got these long chains coming out. Okay, and so I mentioned that they don't always just stick out to one side. They fold over or they go in different directions. So this blue one is saturated. And the green one here is unsaturated at this point. But then this group doesn't have that long chain of carbons. It's got phosphorus and nitrogen in it. So this is a phosphate group. And notice that has a whole bunch of oxygen right in here. And so this part, and then this nitrogen has a positive charge on it too. This makes it polar just in that small area. Okay, so when we have most of it is nonpolar, but we've got a small area that's accessible that is nonpolar, I mean, a small part that is polar, we call that an emulsifier. Okay, and we'll talk about emulsifiers with proteins as well. Okay, so I think that was all I wanted to say about lipids. So in summary, most of our lipids that we call oils and fats are triglycerides. We call them nonpolar, which means they don't mix well with water. The oils are what we call the liquid form. The fats are what we call the solid form. Otherwise, they're pretty much the same. And they're about 9 calories per gram. We're not going to worry too much about the calories right now. We'll talk about energy in a later chapter. But this is more than twice what proteins and carbohydrates are. So if there's a lot of energy density, you can have a little bit of fat and have a lot of calories, which if you are an animal trying to survive the winter, that's great. But if you're trying to lose weight, then fats can be problematic. All right, anybody still need this slide? Yes. Yes.
So the questions that have like the choose where this makes this group, could you explain that a little better? Like how this like to remember?
So you're looking for oxygen because they're usually a functional group, right? So the functional group that has one oxygen and a hydrogen, that's an alcohol, and you can click anywhere on the OH. And if it has 2 oxygens connected to the same carbon, 2 in the same place, then it's going to be a carboxylic acid. So let's back up. So here, this corner is a carbon, and the carbon is attached to an oxygen here and an oxygen here, right? One of them is double bonded. This is a carboxylic acid. So anywhere in this area, you would be able to click on.
Just the others.
Yeah, so technically the carbon, the oxygen, and the hydrogen, all of this is the carboxylic acid, but anywhere that you click in there, it should give you credit for. And if it doesn't, then just let me know. And then on this one, the H is kind of chopped off. This carbon is attached to an OH. So technically all of this is, but I would expect you to probably click on the OH portion. Okay, so the next thing we're going to talk about then is the proteins, and they're probably the most complicated. And so we're just going to go basics right now, just general proteins, and then later on we'll get into a little bit more details on them. Okay, so proteins start with amino acids. We mentioned this when we were doing kind of the overview of the different groups. And so an amino acid has the word acid in it. Notice it has this carbon, 2 oxygens on it. So this is the acid part of it. And then our third functional group is the amine right here. So nitrogen attached to some hydrogens, but mostly you're just looking for nitrogen. If you see nitrogen, that's going to be part of an amine group. So proteins, just like carbohydrates are made of, the longer ones, the polysaccharides are made up of long chains of glucose. Proteins are going to be made up of long chains of amino acids. So we're going to have, this is like a letter that spells out a really long word. Now with carbohydrates, there's really one letter you use a whole bunch, glucose, and then there are a couple other letters that sometimes get used. With proteins, there are about 20 different amino acids. And what makes them different is this thing that says R here. So this carbon in the middle, so it's labeled the alpha carbon, has a carboxylic acid, an amine, a hydrogen, and R, because carbon likes to bond to 4 things. So it's the nitrogen, the carboxylic acid, the hydrogen, and R. R is the rest of the molecule, or sometimes we call it a side chain. And it can be a lot of different things. And so depending on what that is, that makes it whatever word there is, I'm sorry, whatever letter there is in the long word of the protein. So when we have polymers, a lot of times we think of plastics with polymers, but like proteins and starch, those are also polymers. We can have natural polymers. We have the individual unit is called a monomer. So the monosaccharide was the monomer of the carbohydrate. Here, the monomer is the amino acid. All right, anybody still need this?
Which one is the amine?
The amine is where the nitrogen is. I like to throw in these little pictures sometimes. Okay, so what do you call an acid with an attitude? It's an amino acid. There you go.
That was funny.
Thank you. All right, so there are 20 amino acids that we need to build all the proteins that are in our body. Most of them, a little bit more than half of them, our bodies can make themselves. The cells have ways of manufacturing them. There are 9 of them that we can't make. And so we have to get these 9 from our bodies. You do not need to know the names. You do not need to draw these structures. I just want to take a minute to look at them. They all have this top part here is identical. So you probably can't see the top part, but it has a carbon, the acid to the right-hand side, and the nitrogen to the left-hand side. The amine to the left-hand side. So the only thing that's different on these is what's dangling down on the bottom. That's the R group. And so sometimes we have, this is a bunch of carbons in a ring. Here's another couple of rings together. And then sometimes it's just carbon. So this one here, it's just carbon and hydrogen. This one's just carbon and hydrogen. And then sometimes there are functional groups within that. And so we have, let's see, an alcohol here. And this one has an amine here because there's a nitrogen. So there's an OH on the bottom of this one. There's an NH2 on the bottom of that one. So when we have either the alcohol or the amine, or sometimes there's a carboxylic acid, a second one, whenever we have those are polar parts. And when it's all carbon, like this one and this one and this one, then it's nonpolar. So what that means is that if we're using each of these as kind of like the letter in the word, then it's kind of like how some letters are vowels and some letters are consonants. And so, you know, the vowel can appear anywhere, the consonants can appear anywhere. And so that means that parts of the protein are polar, parts are nonpolar, but they don't have a big long chain the way fats do. Fats have that long chain that it's all nonpolar. Here it's kind of spotted back and forth, okay? So you get little pockets of polar and nonpolar.
So what are the functional groups of amino acid?
So on every amino acid, there's a carboxylic acid and an amine.
Okay.
And then the side chain can, but doesn't have to have those or an alcohol, or there's some others that might be thrown in there.
Okay. Because I used that, because that's what the text says is the amine and the
Carboxylic acid, yeah.
But
You have to write it as amine
and alcohol.
Oh, then that's wrong. So which question was that?
It says 2D9.
I'll have to go back and check that. You know, I think I had seen that and I thought I had fixed it. Okay, I will make sure you get credit for that. Okay, why aren't you advancing?
I just wanted to make sure there wasn't like two separate functional groups, like in the question wanted to like answer at the same time.
Again, you don't need to know these names, you don't need to know what they look like, the picture is only here to, if you look closely, you can see the top part is identical for all of them, and the only difference is the bottom part. Okay, so proteins like fatty acids or like phospholipids can be emulsifiers. Okay, so remember that the amines and the carboxylic acids are polar, but that's how we join those together. So that kind of gets, remember we said if that polar area is accessible, it gets non-accessible because it ends up being part of that long chain. The side chains that are mostly carbon are going to be non-polar, and then some of them are going to have nitrogen, sulfur, or oxygen. So if it has any of those functional groups that we're aware of, or if it has, you know, some weird looking thing that has a sulfur in it, that's going to be polar. And so what that means is that while fatty acids are largely nonpolar with just a little bit of a polar end, proteins have pockets of polar and nonpolar. And so what that means is the kind of nonpolar things they can dissolve is going to be different. So water is a pretty small molecule, so it's going to, you know, get its way into the spots regardless. Both of them have small polar parts, but fats tend to have those longer nonpolar parts. So they're going to mix well with fatty acids or with phospholipids, things that also have really long nonpolar parts. But other things can be nonpolar, and in particular air. So nitrogen as a gas is nonpolar, oxygen as a gas is nonpolar, and that's because it's not attached to other things. So remember we talked about the electronegativity difference between carbon and oxygen is what creates the polarity. But when it's oxygen gas, it's just oxygen and oxygen. So that's like identical twins. So they're going to be equally pulling on the electrons, and so it's nonpolar. So proteins are really good at trapping air, whereas fats are better at trapping other fats.
What do you mean by side chain?
The side chain is the part of the amino acid that was represented by the R. That's the thing that can be different from each. The rest of the molecule. Yeah. The rest of the molecule is the side chain. We're going to answer this question. Amino acids have two polar functional groups. The side chains can be nonpolar, so both polar and nonpolar. So that means the proteins tend to be what kind of molecule? So this is just on the last slide, hopefully is right in your notes. Multiple choice. If you can't get the clicker thing to work, you can write it on the whiteboard and show it to me. Okay, so yes, it is an emulsifier. So that is a good word to know. So when we string these together, poly means many. So this is a polypeptide. So the word peptide is a chemical name for the bond between the amine on one amino acid and the carboxylic acid on the other. So that's just where the peptide comes from. So when you hear peptide, think protein. Those start with P, okay? So these are all strung together, these amino acids, and the bond between them is a peptide bond. All right, so a polypeptide is a string of an amino acid, and those are the words that we make up with the letters of the alphabet. And in fact, biochemists use a particular letter for each amino acid. There's 20 of them, so we have more than 20 letters, and that works. And Dr. Hughes, one of our other chemistry professors, will take a word of the day from her word of the day calendar, put it on the board, and then the students have to draw out, based on the letters that are in that word, what the amino acid chain or polypeptide would look like. So you can make literal words out of the proteins. But we're not going to do that. Okay, so proteins, we said, were more complex than the other molecules. So they have 4 levels of structure. The first level, the primary structure, is just the order of amino acids, how we spell out that word. Now, because we have some parts that are polar and some parts that are non-polar, and this is a long chain that can kind of twist around, there are going to be some parts of that that are going to be attracted to itself. So where it gets attracted to itself, it tends to form 2 things. One of them is this corkscrew thing here. So there's like amino acids all along here. And that corkscrew is called an alpha helix. You don't need to know alpha helix. And then the other thing it can do is do kind of a folding back and forth pattern. And so those are called beta sheets. Okay, so our primary structure is just the order of the amino acid. Our secondary structure is that has little coils and folded over portions. We're not too concerned about the secondary structure. It's kind of an intermediate between the first and the third. So the third level is called tertiary, and the fourth level is called quaternary. So the third level is when the whole protein folds up. And it turns out that proteins, the same protein, will always fold the same way. It's not like when you have a piece of yarn and it gets tangled in a different way each time. Every time I have the same sequence of amino acids, it's going to fold up the same way. So that's its tertiary structure. And then sometimes, not all the time, but sometimes those tertiary structures, so here's one, here's one, here's one, and here's one, sometimes those will group together. And so you have a group of proteins called, and that's called quaternary structure. And so the reason I point that out is one of the most important ones is hemoglobin. Have you heard of hemoglobin?
Yeah, I have enough of it to turn blood.
Yep, that happens to me too. So hemoglobin is where the iron sits in your blood cells, and that's what transports oxygen from place to place. And so it's actually made up of 4 proteins connected together. I'm sorry, say again. I don't think I'm going to put primary, tertiary, and quaternary structure on the exam, but it will come back again throughout the semester. Check that. I might put primary structure on. You should know primary is the order of the Okay, so proteins, as I said, have fewer calories than fats. So about 3.5, kind of depends on the protein. And they can be found both in plants and in animals. And they do a lot of things. One of the things they do is control the expression of genes. So your genes basically make proteins. And as they make proteins, those proteins are then used to determine your eye color, determine how much you're going to grow, so whether you're tall or short, determine if you have some genetic disease or don't have some genetic disease. So all of the things that make you come from proteins. All right, so proteins in food, we said they can come from a variety of places. One of the places we primarily think about proteins is in eggs, and we can actually use the white part of the egg called the albumin. We can use that in a variety of ways in cooking, and we can denature it. So what does it mean to denature a protein? Everybody done with this slide? No. Okay.
The brain part didn't monitor, say, single amino acids.
Oh, you're answering my question. So that's a good guess, but no. This is where having the primary, secondary, tertiary structure is important. So basically, it leaves the primary structure in place. So we still have the individual amino acids, but it unfolds it. So it takes away the tertiary and sometimes even the secondary structure. So instead of it being scrunched up into a ball, it unfolds. Okay, so that's what denaturing is. Often when we denature, since we're exposing parts that weren't exposed before, two proteins can get tangled together. And when we clump together those proteins, we call that coagulation. Oh, I forgot this, okay. I talked about these, but you might want to have these in your notes. So some of the functions, we said regulation. So regulation is determining how fast your metabolism goes and whether or not you need to have more insulin in your blood, those kinds of things. We said some hormones come from steroids, other hormones come from proteins. So there are two different kinds of hormones. What are antibodies used for? Fight off Russians,
Viruses.
viruses, or bacteria, or whatever. No offense to any Russians in the room. So yeah, antibodies fight off diseases, basically. And so we use proteins to do that. And then, of course, our muscles are made out of proteins. So there's lots and lots of functions is the main idea. Yes,
You said the two hormones, steroids, what else?
and proteins. So the sex hormones we talked about, estrogen, testosterone, progesterone, those ones that you know the names of, those are going to be the steroids. But you've probably heard of insulin, right? Insulin is also a hormone and it's a protein. All right, so one of the ways that your body uses proteins to regulate things is by enzymes. So enzymes are proteins, but they're just one group of proteins. And these are things that serve as catalysts. So in general, in chemistry, the word catalyst means that you are speeding up a reaction. So an enzyme speeds up a biological reaction. And these are all names of enzymes. Lactase breaks apart lactose. Amylase breaks apart amylose. Tyrosinase, I'm not sure how to say it, breaks apart the amino acid tyrosinine. So what do you notice that it has in common? They all have ACE. So just like OSE usually means that it's a sugar or a carbohydrate, ASE usually means it's an enzyme. So all of the processes that make you a human would take millions of years without a catalyst. And with the catalyst, those things are happening thousands of times per second. And so some things that it might do is help grow food, help the food ripen, and also cause the food to spoil. So we do need to know the word enzyme. We need to know that it is a protein, and we need to know what the word catalyst means, that it speeds up a reaction and that enzymes are catalysts. Make connections between enzyme, catalyst, and protein. Okay. What role do enzymes play in chemical reactions? You can give me a word for what they do. What was the one word that we said enzymes are this? Thought I had a multiple choice on this question. Last one. Yes. One more time. Anybody needs to show me their word? All right, so all of this stuff at the top here is right catalysts, or it's a catalyst and it speeds up reactions. They are special proteins, they speed things up. One of the things they do is growth, but that's not all enzymes. Some enzymes do different things. So generally, enzymes are catalysts that speed things up. So destroying proteins or denaturing proteins. Denaturing means we unfold the proteins. So we might just take away that tertiary structure of it all being bunched up, or we might actually unscrew the corkscrew or unfold the beta sheets. And then coagulating is when different proteins join together. So I started talking about eggs, and we know that for a raw egg, the egg white, in quotes, actually isn't white, it's clear and colorless, maybe slightly off color, maybe it's a little bit yellow or something. But basically it doesn't have a color. But when we cook it, it turns into something white. So by cooking the white part, we are denaturing it. And so we can look at different things that can turn the white part of the egg to actually be white instead of clear. And so one of the things is with heat. So you can fry an egg, and that's going to turn the egg white. Or you could hard boil the egg, or you could poach the egg. Any of those things that add heat. Okay. If you've ever had, oh yeah, go ahead, Brian.
On the homework, I put heat, acid, and physical in account if that's wrong, would that be the same or not?
Well, yeah, because it was like straight up.
Did you
I didn't say
type it in?
chemical. Yeah, this was typed, it wasn't.
It was just that I didn't guess one of the hundreds of ways you could have written it. I'll go back and check that. Okay, so heat is one way. Now, some people enjoy eating, I've never actually eaten one because it sounds gross to me, pickled eggs. Have you ever heard of pickled eggs? So basically the idea is they put the raw egg into an acid, pickle juice, lime juice, vinegar, something like that. And the acid seeps in and reacts with the proteins, and that causes the proteins to denature and coagulate. So this is how we can denature a protein, is with chemicals, often acids. And then the other thing we can do with an egg white to make it look bright white is to whip it. So if you've ever seen somebody make a meringue, they start with the liquidy, colorless egg white, runny stuff, and they whip it, and then after a while it becomes a foam, and that's where it being an emulsifier is important, because as the protein unfolds, the nonpolar regions trap air into those little pockets, and that's what makes it into the foam that it is. But you're also unfolding those proteins, and then that action is causing them to kind of coagulate and join together. Okay, so the denaturing is the unfolding, and coagulating is joining together. And so I have next to heat, this is why we have fevers, because when you have some kind of a disease, one of the ways your body attacks that disease is to try to denature its proteins, or to cause its proteins to coagulate. So your body temperature rises to make the proteins of the bacteria or whatever malformed so that it can't do what it's trying to do.
The denaturing question on the homework was another one where like you had to put it a specific way.
Mhm. Yeah, so all the ones you have to type in, I go back and check those by hand. I just haven't done it yet because I knew that some people might need to go back and look at the project. Okay, so I think that was the last of that. Now there are. Okay, let's try this one. Which biomolecule is the largest and most complex? We've been talking a lot about it today. So more time. Yay, everybody's correct. Okay, now just remember that I might ask a similar question on your test on Thursday, so remember it that long. Okay, think back. Let's see. Well, this is a good one to know. Which functional group is found on proteins, but not on the other biomolecules? Those are new functional group that we talked about today. And that was the amine, that was the new one for today. So we did talk about carboxylic acids on fatty acids last Thursday. Okay, so what I want to do is I want to take a look from the chapter of few images of the natured proteins. And this is towards the end of the chapter. Okay, so this is an egg that has been pickled. It's been sitting in vinegar. And so the, you got little bubbles in here, but the membrane is kind of holding it all together. And then the white part isn't quite as white as when you cook it. And that kind of depends on how long you have it sitting in the, what do you call it, in the vinegar or the acid. Okay, so there's the pickled egg. And then the next picture is showing a meringue, which is made by stress. Whipping the egg whites causes them to become white and stiffer, but not rubber. So those are two ways that we are adding stress: physical stress, chemical stress, heat stress. And that can denature the proteins. And it doesn't just work on egg whites, it's just easiest to see with egg whites. Okay. Questions about proteins?